This project attempts to relate the organophosphate concentrations in California surface water as a function of California agriculture. This project was completed with the assistance of the Geospatial Analysis Lab at the University of San Francisco and with Professor David Sa. The basis for this project is in the International Agency for Research on Cancer's recent reclassification of 5 pesticides to different carcinogenic groups. Glyphosate, MALATHION, and DIAZINON were classified to 2A, TETRACHLORVINPHOS, PARATHION were classified to Group 2B. The first 2A chemical, glyphosate, is a herbicide that is applied directly to leaves. It works via inhibition of the shikimic acid pathway and is used very commonly with agriculture and residential applications. You can see in the map of California that there's a heat map regarding sampling density, not sampling frequency. The first 2A pesticide is malathion. Malathion, like all organophosphate insecticides, works via inhibition of acetylcholinesterase, causing toxicity by buildup of acetylcholine at neuronal junctions. Diazinon is the last 2A pesticide and is used as an insecticide, acaricide, and nematicide. Parathion, also found as methylparathion, is similar to malathion and is the first 2B insecticide. TETRACHLORVINPHOS is the last 2B pesticide and is the least used of all 5 chemicals. The geographical extent of this study is limited to California. California has the largest agricultural output in the country, as well as stringent pesticide regulations which lead to excellent record keeping. All geographic information is taken from the United States Census Bureau Tiger files. The main challenge of this project was integrating 3 large and different datasets. The first, the USGS National Hydrography Dataset, is a network layer of all surface water features down to a very detailed level. The second, the U.S. Department of Agriculture Crop Data Layer, is a raster dataset generated from satellite imagery. It shows over 200 different kinds of crops based on their spectrums. The third dataset is from CDN, or the California Environmental Data Exchange Network, and is a user-queried database of pesticide surface water concentrations that is geolocated with latitude and longitude. The first step in the GIS analysis was to take the crop data layer, extract it to just the state of California, and then reclassify the crop values to more general ideas. For instance, several types of rice would be classified into a rice category, different kinds of grains would be classified into a grains category. The next step from there was to use segment mean shift to reclassify the raster in a simpler manner based on neighboring cells. We then converted the raster to a polygon. The second step in the GIS analysis was to simplify the National Hydrography dataset. As is, it was too large to manipulate cleanly. We took the sampling incident locations, created a buffer around them, intersected the buffer with the dataset, and then clipped the buffer to the dataset. That reduced the amount of data we had by about 2/3 and made the dataset far more manageable. On this map, you see the simplified and processed crop data layer over the state of California. There are 17 crop categories down from the approximately 200 that were exist previously. In the zoomed in section, you can see the addition of the National Hydrography dataset to the cropland cover. The final step in our GIS analysis was to create a network. Using the National Hydrography dataset as the network and loading those sampling locations as facilities, we generated service areas within 1, 5, 10, and 20 miles of each sampling point. The service areas were then intersected with the crop data layer to get a picture of the amount of crop area at each of the 1, 5, 10, and 20 miles from each sampling location along the network. In this image, you can see a sampling location in yellow, and in black, a full 20-mile buffer along the network. Beige is background, all other colors are different kinds of crops. This image is the same data as in the previous image, albeit visualized differently. Yellow is again the sampling location, beige is again the background. However, the service areas are visualized in yellow, purple, light blue, and dark blue respectively. Green again is crop. Here you can see a Google Earth image of the area of the previous images. It's clearly cropland, and as we zoom out, you can see it is slightly east of Chico and west of Orland. The latitude and longitude is approximately 39 by negative 121. Due to the extensive nature of the results, we only present parathion as an example. You can see Given the crop at the bottom, cotton and grass are the most indicative of a positive parathion sample. However, there is very little cumulative data for the first 1 to 5 mile buffers, and most cropland appears to be within 10 to 20 miles. This next graph is the same data again visualized separately. Instead of a cumulative buffer, this is individual buffers, and the results are not summed. It appears that extreme proximity to a cotton or grass field is not the main driver of whether or not there is a parathion sample, but simply the cumulative as expressed in the previous graph. This project was completed using data from the USGS National Hydrography Set, the California Environmental Data Exchange Network, the USDA Crop Data Layer, U.S. Census Bureau Tiger Files, National Pesticide Information Center fact sheets, Wikipedia for chemical structures, and Google Maps for map and satellite images. For further information, please read the white paper or contact Andrew Patton at anpatton@usfca.edu.